The DNA. You've probably heard about DNA countless times. But what exactly is it? Let's dive deeper and discover more about DNA in this video. DNA is the master code of life, silently scripting every trait, feature, and function that makes you uniquely you. DNA, or deoxyribonucleic acid, is a long polymer made of repeating units called nucleotides. It is the molecule responsible for storing genetic information in almost all living organisms. DNA provides the instruction for building and maintaining cells, and it is passed down from parents to offspring. DNA is found in the nucleus of cells in humans and many other organisms. Length of DNA. Another fascinating aspect of DNA is its length. The number of nucleotides and base pairs plays a key role in determining the overall length of a DNA strand. You can see on the left side is the image of nucleotide and on the right side is the image of base pair. We will discuss more about them in the later part of this video. Here's a list of DNA lengths for various organisms mentioned in the NCERT. Now, you might have noticed that in bacteriophage 5X174, length is in nucleotides, indicating that the organisms have single-stranded genome, whereas in bacteriophage lambda, E. coli, and human have their length in base pairs, indicating that the organisms have double-stranded genome. You might be curious why the DNA length varies from one organism to another. The length of DNA varies from organism to organism primarily due to the complexity and genetic requirements of each species. Now let's look into DNA from a historical perspective. Number 1. Friedrich Miescher. Friedrich Miescher identified a molecule inside the nucleus of white blood cells and termed it nuclein in 1869, laying the foundation for the discovery of DNA. Number 2. Erwin Chargaff. Erwin Chargaff's rule states that in double-stranded DNA, the amount of adenine always equals the amount of thymine and the amount of guanine always equals the amount of cytosine. Number 3. Rosalind Franklin and Maurice Wilkins. In the early 1950s, Rosalind Franklin and Maurice Wilkins used X-ray diffraction to capture images of DNA fibers. Franklin's famous Photo 51 provided crucial insight into the helical structure of DNA, although her contributions were not fully recognized at that time. Number 4. James Watson and Francis Crick. In 1953, James Watson and Francis Crick, using Franklin's data and their own insights, proposed the double helix structure of DNA. Next, we will discuss about components of DNA. A nucleotide is composed of a phosphate group, pentose sugar, and a nitrogenous base. Number 1. A pentose sugar. Two types of sugar are present, deoxyribose in case of DNA and ribose in case of RNA. Among the pentose sugar, deoxyribose is more stable compared to the ribose sugar. Number 2. A nitrogenous base. It is a nitrogen-containing molecule having similar properties as of base. There are two types of nitrogenous base. Purines consist of adenine and guanine, and pyrimidines consist of cytosine, uracil, and thymine. Thymine is present in DNA and uracil is present in RNA. The rest bases are common for both DNA as well as RNA. A nucleoside is formed when a nitrogen base is linked to pentose sugar through N-glycosidic bond. Refer to this table for the various types of nucleosides that can be formed. Number 3. A phosphate group. When phosphate group binds to the 5' OH of nucleoside through phosphoester bond, forms the nucleotide. Two nucleotides join together through phosphodiester linkage, and in similar fashion n number of nucleotides join to form polynucleotide. Now, after the polynucleotide chain is formed, it has two ends, 5' end and 3' end. The end having free phosphate group is marked as 5' end of polynucleotide chain, while the other end having free hydroxyl group marked as 3' end of polynucleotide chain. Watson and Crick model of DNA, also known as double helix model, describes DNA as a twisted ladder. The sides of the ladder are formed by sugars and phosphate groups. This backbone provides structural support and stability to the DNA molecule. The nitrogen bases are projected inwards. The bases of two strands are paired through the hydrogen bond to form base pairs. Adenine pairs with thymine through two hydrogen bonds. Guanine pairs with cytosine through three hydrogen bonds. Now, here's the question, why purines pairs with pyrimidine? As you know that purines are double ring structures and pyrimidines are single ring structures, so for the DNA helix to maintain a consistent width, a purine always pairs with a pyrimidine. The two strands of DNA run in opposite directions, known as anti-parallel orientation, in which one strand run in 5' to 3' direction, whereas the other strand run in 3' to 5' direction. The two strands are coiled in a right-handed fashion. Each turn of the DNA helix is called the pitch, and it is about 3.4 nanometer. And one turn of DNA consists of approximately 10 base pairs. The distance between base pairs in the helix is about 0.34 nanometer. Questions to explore more. In this section, I encourage you to discover the answers to the question on your own. How does DNA structure allow it to hold so much information in such a small space? Why does DNA function as the genetic material in most organisms and not RNA? What are genes and how do they regulate function in our body? How is genetic information transferred from one generation to the next? Thanks for watching this video. This video was presented by Shrushti Purohit under the guidance of Dr. Gopalji Gopal sir. I hope this video sparked your curiosity about DNA. If you want to dive deeper into genetics, evolution, or any other fascinating topics, be sure to hit the like button, subscribe, and turn on notifications so you don't miss out on future videos. Have any questions or want to explore something further? Drop them in the comments. I would love to hear your thoughts. Till then, keep exploring and stay curious.